line on that, okay? Now here comes the question you might ask. Why does the RNA contain the uracil base and not the thymine? It contained the other three the same as, you see, these are the same as these. But the RNA has the uracil, the DNA has the thymine. How does it get there? Well, in that transcription process, what that RNA polymerase does, it brings along a nucleotide, which is a structure composed of a phosphate with a ribose sugar and a base. The base in that nucleotide is uracil. And you say, well, it's just floating around out there. Well, really it is, but it's not just there accidentally, right? It's there for a reason. And what it does is the polymerase maneuvers it into the reaction position for the RNA to pluck that uracil, pluck that thymine off of that where the DNA had it and put the uracil in place of it. It's a substitution, okay? And these are the basic reasons, apparently, as far as we know now, why this is true. First of all, the thiamine with that methyl group, okay, can be functional in the way that it has to be as a DNA base, but also protects that particular base in the functional process. Remember, it's located, I don't remember because you don't know, but in the structural form of that DNA double helix, those thiamines are the most vulnerable ones. They're the ones sticking out. They're the most vulnerable to alternate attacks. And if they get attacked and, of course, reacted upon, what's going to happen? You're going to have a mutation of the DNA, right? So you don't want that to happen if you want the integrity of that DNA to remain, right? So the methyl group helps to protect that situation. That one base has that one unit that makes a lot of difference in how the DNA is going to survive. The second thing is uracil itself has a much greater affinity to base pair with adenine. Now, of course, it's not a double helix, but it will undergo base pairing with other units because all those bases on RNA can base pair just as well as they do on DNA. And since it has a greater affinity to base pair with adenine, then that mechanism is crucial for RNA's function to be carried out. Because if it didn't do that, the RNA wouldn't function properly. See? See the design going on here with all this stuff? It's all chemistry, but guess what? It's not a haphazard chemistry. It's a very critical design here. The third thing, there are repair mechanisms in DNA, as everyone knows. If that didn't happen, we'd be in bad shape because our cells would be undergoing destruction all the time, wouldn't they? But there's self-repair going on. And the repair enzymes, notice that word enzymes comes in, they're critical. They find it easier to recognize that DNA if that thymine has that methyl group on it. Now don't ask me why. That's just the nature of things, the way God designed it. It makes a difference, and that's why it's there. So the DNA repair mechanism requires that thymine to be there and nothing else. And then fourthly, uracil, that's on RNA, that molecule gets converted or replaced to cytosine, which could lead to other things, but what it basically does is it just cuts off the function of RNA when it has to be cut off at the right time. Control all the way along in these things. Tremendous amount of control. Okay, so let's move on. Examples of complexity. The human genome. I'm sure you've heard about this. Has about 6 billion base pairs. Here now we're talking about in the DNA structure, right? With 2 strands of the double helix in the DNA. The code for countless numbers of RNAs. Now remember, those RNAs are coded from the DNA structures, right? So they code for countless numbers of RNAs and some 200,000 different proteins. That's what the genome does. And in this case, it would be the example of the human genome, but all genomes do that kind of thing. That's phenomenal. I mean, when you think of the number of entities that are involved here, I mean, they're almost countless. They're phenomenal. And they're all bottled up in these little bitty small regions that you can barely see with the microscope. You know, you can see them with high resolution microscope, but it's amazing. It's just totally mind-boggling. Then we come to the RNA, the ribonucleic acid. I'm going to focus on that a little bit. It's the single strand, as we said, with the phosphate and the ribose and so on. And the one base is different. We all know that already, right? Okay, so we already went through that. Here's a little bit more of that critical interaction in the double helix. You notice the adenine communicating over here with the thymine. It does that by a special kind of chemical bonding called hydrogen bonding. And the same thing does with guanine and cytosine, and with cytosine and guanine, and then here with thymine and adenine. Now, if you change the order of those things, from what that example shows there, if you change the order of those things, you change the function completely. If you change the order of one of these pairs and reverse it, you change the function completely. It's just amazing. It's so critically locked in. You would change the function completely. So it's that critically balanced that we have to keep in mind here. Now, what are the special features of RNA? Well, special features of RNA are, number one, you got messenger RNA. It's the one that carries the genetic code to form the specific amino acids for producing a protein. That's its function as an RNA, the messenger RNA that comes out of the transcription process. Now, the next thing you've got is a transfer RNA. It transfers, as the name says, specific amino acids to be coded within a unit of the cell called the ribosomes, which make the proteins. In other words, it's the factory in that portion of the cell that generates the proteins. It's manufacturing proteins. And the transfer RNA is involved in that process. No transfer RNA, no proteins. Okay? Then the ribosomal RNA, and really about 90% is ribosomal, interestingly enough. That only leaves 10% for these other two, right? But that catalyzes processes of protein formation for amino acids. Now, that's very critical. Catalyst means what?